This protocol improves the basic concept of wet spinning to develop a material from a single fiber shape into various shapes that can be used for any preferred medical application.
This technique is simple, inexpensive, and does not require the addition of synthetic polymers for customization of the natural polymers into a rich source of protein for materials of interest.
This technique can be extended to biomaterial development for clinical therapy, such as central or peripheral nerve system treatment, blood vessel replacement, or direct urinary tract reconstruction.
This method is intended for medical application, including as a strategy for tissue regeneration or regenerative medicine.
This technique can be easily implemented by beginners. Note that after several molding repetitions, the quality of the solution should always be checked to maintain an optimal formation.
To form the wet spinning tube, first submerge a 24-gauge by 3/4 inch peripheral venous catheter into freshly prepared gelatin solution for 3 seconds, followed by immersion in freshly prepared acetone solution for 1 minute. Repeat these steps 20 times. After the last acetone immersion, let the molded tube dry for 5 minutes at room temperature, and use a hemostat to carefully transfer the gelatin tube from the catheter